Glycolysis. Glycolysis is the first step in cellular respiration and it occurs in the cytoplasm of the cell. The 10 steps of glycolysis are divided into 2 phases. The first 5 are preparatory phases and the last 5 are payoff phases. Step 1. The enzyme hexokinase removes a phosphate group from an adenosine triphosphate molecule to make it adenosine diphosphate, then adds it to the glucose molecule to form glucose 6-phosphate. This makes the glucose more reactive and the added phosphate group prevents it from crossing the cell membrane, trapping the glucose inside the cell to be metabolized. Step 2. Phosphosexose isomerase rearranges some of the hydrogen and oxygen atoms in glucose 6-phosphate to make it into an isomer of itself, called fructose 6-phosphate. Step 3. This rearrangement allows phosphofructokinase 1 to transfer a phosphate group from another ATP molecule to the fructose 6-phosphate to produce fructose 1,6-bisphosphate. Magnesium is a cofactor of this process and it is irreversible. Step 4. The enzyme fructose bisphosphate adylase breaks up fructose 1,6-bisphosphate into 2 3-carbon sugars that are isomers of each other, dihydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate, also called GAP. However, only GAP can proceed to the next step. Therefore, in step 5, DHAP is converted into GAP with the help of an enzyme called triose phosphate isomerase. So far, this process has used up 2 ATP molecules, converting them to ADP and produced 2 3-carbon sugars, each with a phosphate group. The next 5 stages use these sugars to get the payoff. Remember that 2 GAP molecules are made, so the following reactions happen twice. Step 6. The GAP molecule is oxidized with the help of the enzyme glyceraldehyde 3-phosphate dehydrogenase, and NAD+ is reduced to NADH and H+. This stage releases energy, which is used to add another phosphate group to the GAP molecule, creating 1,3-bisphosphoglycerate. Step 7. The enzyme phosphoglycerate kinase then transfers one of those phosphate groups from 1,3-bisphosphoglycerate to ADP, forming an ATP molecule and 3-phosphoglycerate. Step 8. Phosphoglycerate mutase then relocates the remaining phosphate group from the 3rd to 2nd carbon atom, isomerizing 3-phosphoglycerate to 2-phosphoglycerate. Step 9. 2-phosphoglycerate loses a molecule of water with the help of the enzyme enolase, becoming phosphenylpyruvate or PEP. This is an unstable molecule and is primed to lose its phosphate group. Step 10. Pyruvate kinase is the enzyme that helps along this process. PEP readily gives up its phosphate group to an ADP molecule, forming ATP, and is converted into pyruvate, the final product of glycolysis. So, this process has produced 2 pyruvate molecules, 2 NADH molecules, and 4 ATP molecules. However, because the first stage of glycolysis used 2 ATPs, we have a net production of 2 ATP molecules. We hope you have enjoyed our presentation and now have a better understanding of how glycolysis works. Thanks for watching.